Cytology, the study of cells. Cytology is the study of cells. Referring back to the levels of organizations, cells are the first things that are considered to be living. There are two parts to a cell, the cell membrane and the cytoplasm. The membrane surrounds the cell, and the cytoplasm includes everything inside the cell. The cytoplasm is further divided into two components, the cytosol and organelles. The cytosol is the fluid in the cell, and the organelles are the functioning components. Cells come in a variety of shapes. Squamous cells are flat and thin. Polygonal cells are irregularly shaped, and they're not quite round, but are often square and a little bit asymmetrical. Stellate cells are star-shaped. Cuboidal cells are shaped like a cube. They are roughly the same height and width. Even though they are cuboidal in shape, they sometimes have rounded corners and can look a little bit like circles. The key is that they are the same height and width. Columnar cells are tall and thin or rectangular. They are taller than they are wide. Spheroid cells are round or oval. Discoid cells are disc-shaped. Fusiform cells are thick in the middle and thin at the ends. And fibrous cells are long and thread-like. The cell membrane, or plasma membrane, has a number of functions. For starters, communication is important. This membrane allows cells to communicate with other cells. The cell membrane also holds the cell shape and functions in protection. The cell membrane maintains an electrochemical gradient. There are differences in ion concentrations between the inside and outside of cells. Cell membranes are semi-permeable or selectively permeable. This means that membranes allow some things to enter or leave the cells, but prohibits other things from entering or leaving the cell. A cell membrane is formed from two layers of phospholipids, sometimes referred to as the phospholipid bilayer. Each individual component, the phospholipid, will have two parts, a hydrophobic portion and a hydrophilic portion. The hydrophilic portion of the molecule will be the phosphate heads that can interact with water, and the hydrophobic portion will be the fatty acids that have an affinity for other fats more so than water. The extracellular fluid and the intracellular fluid both contain water, so the phospholipids orient themselves so that the heads are facing to the outside and the inside. All the tails point to the center of the membrane, where they are closer to each other. Phospholipids do not attach to each other. They are more independent and can move separately. This allows the membrane to be fluid and flexible. Although it's advantageous for the membrane to adapt its shape, we don't want it to be too malleable. This is where cholesterol is sometimes important by providing stability to different components of the membrane. There are also a number of proteins associated with cell membranes. Membrane proteins often act as receptors for communication. They often serve as second messengers. They work as enzymes for various processes such as digestion, breaking down hormones, and signaling. And they may also form transportation channels and act as carriers for molecules that need assistance to get into and out of the cell. They are also essential for cellular ID. Proteins on the surface of a cell allow cells to recognize similar cells. Last but not least, cell membrane proteins help cells attach to their neighbors. Here's a picture of a phospholipid. The round hydrophilic head is formed from phosphates, hence the name, and the tails are made from fatty acids, which are long chains of carbon and hydrogen. Here's the diagram of the entire membrane. You can see the cholesterol molecules and the proteins. Also notice that the phosphate heads form the internal and external surfaces. Since the heads are hydrophilic and attracted to water, they line up on the outside and the inside, and the tails face the center of the bilayer membrane. As mentioned, due to the membrane structure, some things pass more easily into or out of the cell than others. Small things such as water, oxygen, and carbon dioxide can slip easily between the phospholipids, without any problem. Also, non-polar molecules, for example like carbon dioxide, and hydrophobic molecules like lipids can also pass through the phospholipid membrane. Substances that are more similar to phospholipids will meld with the membrane and pass through much more easily than substances that are chemically different. Substances that have a harder time passing through are oftentimes large in size or have an electronic charge. These molecules often need help from channels or carrier proteins to get into or out of the cell. Many important molecules and substances must be able to move into and out of cells. There are two ways to categorize membrane transport: passive transport mechanisms and carrier-mediated transport mechanisms. Passive transport does not require adenosine triphosphate or energy. There are a few mechanisms that are important to know about. Filtration is transport that uses hydrostatic pressure to move particles. Pressure from water drives water and other particles through cells or through membranes. Simple diffusion is the movement of particles down a concentration gradient from high concentrations to low concentrations. Think about what happens when someone sprays perfume. It's rather potent right away near the site where it's sprayed, but given some time, the scent will diffuse through the room. Diffusion is influenced by a number of factors. For starters, temperature. Increase the temperature and you will increase the rate of diffusion. Molecular weight. Heavy particles do not move as fast as smaller particles, which are slightly more rapid. The steepness of the gradient is also important. If there is not much of a difference between two different regions, diffusion will be slower. Membrane surface area. The larger the surface area, the more locations there will be for things to cross the membrane. Finally, membrane permeability. Some substances can pass easily and others do not pass as easily. Finally, osmosis is the diffusion of water. It's defined as the movement of water from areas of high water concentration to areas of low water concentration. Carrier-mediated transport involves proteins found in the cell membrane. There are two types of carrier-mediated transport that we need to focus on. The first is facilitated diffusion. Some substances are often too large to flow across the membrane without any help. The membrane uses proteins to help these particles move across the membrane. This movement is still considered passive because it is down the concentration gradient from areas of high to low concentration. Active transport is the second form of carrier-mediated transport. It requires the use of ATP or energy to move particles across the membrane, and it also involves a protein molecule. With active transport, particles move against a concentration gradient or from areas of low to high concentration. This is the opposite of diffusion. Think of paddling up a river. This is a great video that will give you a quick overview of cell membrane structure, osmosis, and active transport. The second part of a cell, the cytoplasm, consists of all of the contents of the cell. Inside we find the cytosol, which is fluid, and organelles. The cytosol is sometimes referred to as the intracellular fluid, or ICF. This is different from the extracellular fluid, or ECF, which includes all of the fluid found outside of the cell membrane. The cytoplasm also contains organelles, the small organ-like structures found within a cell that are not considered to be alive, but that are essential for the functioning of the cell. The nucleus is often the dominant organelle in a cell. There are some cells that we will discuss that do not have a nucleus, but when it is present, it usually dominates a major portion of the cytoplasm. Think of the nucleus as the control center. It holds the DNA, which consists of a series of recipes that will be used to put amino acids into specific sequence. The nucleus itself is surrounded by a bilayer membrane called the nuclear envelope. In addition, the nucleus contains a structure called the nucleolus, which is also used to produce ribosomes, which will be discussed shortly. Here are two views of the nucleus and the nuclear envelope that surrounds it. Inside the nucleus, there is usually a single nucleolus, which is the darker spot. As mentioned previously, the nucleolus will produce another type of organelle called ribosomes, which will be discussed shortly. The endoplasmic reticulum, or ER, is a system of interconnected channels. The channels are furthermore known as cisternae. There are two segments or two portions of ER, rough and smooth. Rough ER is continuous with the nuclear envelope and is primarily responsible for protein and peptide production. Rough endoplasmic reticulum contains many smaller organelles known as ribosomes. Smooth ER is still continuous with the rough endoplasmic reticulum, but functions more to detoxify different chemicals within a cell. Smooth endoplasmic reticulum does not have ribosomes. Here's a picture comparing and contrasting rough endoplasmic reticulum with smooth endoplasmic reticulum. The small dots visible in the rough ER are actually ribosomes. Ribosomes are small organelles that are produced in the nucleolus of a cell. They are usually found in large collections on rough endoplasmic reticulum. Ribosomes are responsible for decoding genetic information and assembling amino acids into short chains called peptides or longer chains called proteins. The Golgi complex, or complexes of a cell, are systems of small cisternae, much like endoplasmic reticulum, but slightly smaller. They tend to resemble stacks of pancakes. The Golgi complex will finish off protein synthesis and also build carbohydrates. When synthesis is finalized, the Golgi complex will help package proteins and sugars into vesicles that can then be moved through the cell. The vesicles look like little tiny bubbles and essentially are transport capsules. Vesicles can also transport things into and out of a cell through processes of exocytosis and endocytosis. Here's a slide showing the cisternae of the Golgi complex and the smaller vesicles that can be seen budding off of the cisternae. Exocytosis occurs when vesicles from inside of the cell fuse with the cell membrane and release the contents to the outside of the cell. Endocytosis occurs when the membrane forms a vesicle around an object or some components of the extracellular fluid to allow it to enter the cell. The membrane folds in and pinches off to create a vesicle that is then pulled into the cytoplasm. Lysosomes are a specialized type of Golgi vesicle that travel around the cell and break down organelles and other wanted particles. White blood cells use lysosomes to break down bacteria inside of a cell, and liver cells use lysosomes to release stored glucose into the blood. Lysosomes are small pockets or bubble-like structures that usually contain digestive enzymes or proteins that are capable of breaking other molecules down. There are about 50 types of proteins found in some lysosomes. Peroxisomes actually resemble lysosomes but are produced by the endoplasmic reticulum rather than a Golgi complex. Peroxisomes detoxify cells by producing hydrogen peroxide that will be used to break down internal components of a cell. Here are some pictures that compare and contrast lysosomes and peroxisomes. Mitochondria are small bean-shaped organelles that have internal folds called cristae. Essentially, mitochondria synthesize adenosine triphosphate. Think of them as the powerhouses for a cell. Mitochondria also have their own DNA. A small ring of DNA is contained and is passed on from the mother to child. So you can see trace lineages of mitochondrial DNA as well as nuclear DNA. One theory regarding why mitochondria has its own DNA is that it was once a single-celled organism that became incorporated into human cells and now serve to function only as an organelle. Here's a visual representation of a couple of mitochondria. You may also notice that ribosomes are found inside of mitochondria. Since mitochondria have their own DNA, they do use ribosomes to produce the corresponding proteins. Centrioles are usually found in pairs in larger regions called centrosomes. They are short cylindrical assemblies of microtubules or small protein rods. They are usually found at right angles or in perpendicular form to one another, and they are used during cell division to help with internal rearrangement. This picture shows two centrioles. The centriole on the left is actually pointed directly towards you, so you're looking down into it. The small orange rods represent microtubules. These microtubules will be used during cell division to help move internal components of a cell. Certain cells oftentimes have projections sticking up off the cell surface. Some common projections include cilia, which are small finger-like structures that always occur in large numbers. Cilia work together to move fluid from one place to another. Another example of a projection is the flagellum, or often the tail of a sperm. A flagellum has the ability to move a sperm cell in a certain direction through fluid. This diagram shows how cilia work together to move mucus and any particles that may be trapped in that mucus in a certain direction. The cytoskeleton inside of a cell is a network of tube-like structures. The cytoskeleton helps determine the shape and provides support and organization for the cell, as well as aiding in movement of cells that have the ability to move. Think of the cytoskeleton like the I-beams or internal support structure. The cytoskeleton is primarily made of proteins, microfilaments, and microtubules. Microfilaments form a skeleton-like structure in the membrane, and the microtubules are a protein cylinder that helps maintain shape and rigidity and also aids in cell division. If you look closely at this diagram, you can see the microfilaments in the internal folds of the plasma membrane. The microtubules are bigger, longer tubes of proteins that radiate out from the center of the cell. There are a few terms regarding cell growth and death that you should be aware of as we move forward. Mitosis is basic cell growth and division. One cell divides to form two identical cells. Meiosis is the cell division process that's used to form sex cells or gametes, more commonly known as egg cells or sperm cells. We will deal more with meiosis during the chapter on the reproductive system. There are a few types of ways in which cells cease to exist. Apoptosis is programmed cell death or the natural death of the cell. Cells, like all different things, have specific lifespans. Different types of cells will have different lifespans, some longer or shorter. Necrosis is cell death due to trauma or infection. This is pathological and not part of the normal cellular lifespan. Autophagy is the breakdown of cellular components. For example, when a lysosome inside of a cell breaks down parts of its mitochondria.